So I'm a microbiologist and I study bacteria. And I'm interested in bacteria and how they interact with their environment. Bacteria are very, very small and they're ubiquitous. They're found everywhere. And they respond by changing their gene expression in response to changes in their environment. They do that in order to be able to grow better or to be able to grow in new environments. And that's been my research for many years. I started thinking about antibiotic resistance as a side or part of my work with my course in microbiology. I started teaching microbiology many years ago, but as the problem of antibiotic resistance became more and more of an issue, I started to include more of that information in my courses, and as such I started to get more and more interested in. At the same time, I started to realize that much of what I was studying about gene regulation and the environment actually applies to the problem of antibiotic resistance. And so there were places where my research connected with the problem. So within CARE, I actually have two roles. The first is involved in education, and the second in research. In education, I'm interested in creating resources that other teachers can use in many different courses. So we're going to create resources that can be used in first-year courses for non-science majors or for master's level science courses, etc. We want to have a little bit of antibiotic resistance available to teachers in many different fields. In research, I'm interested in gene transfer. So when a bacteria becomes resistant, it can do that two ways. One, it can simply make a mutation or be selective for a mutation that allows the cell to grow under that antibiotic stress. But a more common way is that antibiotic resistance ends up on a plasmid, and these are DNA molecules that can be transferred between bacteria. And the result of that is that antibiotic resistance will spread very, very quickly. It can spread within a population that's infecting a person, for example, or even spreading across the world. And so what we're interested in is understanding gene transfer more so that in the long term we can perhaps inhibit gene transfer. And what that would cause is a slower evolution to antibiotic resistance and lessen the need for new antibiotics.